Beautiful people. In this sequence, we will take a look at the notable people in biochemistry. And yes, okay, just a few of them, those that are somewhat related to our course throughout this semester. So yes, let's take a look. The list here is Frederick Wooler, who synthesized an organic compound from an inorganic compound. Because during the time of Wooler, there was this theory stating that, or there was this belief stating that organic compounds are only produced by living organisms. But then, the experiment of Wooler, he synthesized or he produced an organic compound from an inorganic compound. How did he do that? He heated ammonium cyanate. Ammonium cyanate here is an inorganic compound. He heated ammonium cyanate to form urea. Your urea here is an organic compound that is found in the urine and blood. So in his experiment, Frederick Wooler was able to synthesize urea without the use of human kidney. He was able to synthesize urea without the use of human kidney. And accordingly, this experiment laid the foundation to modern biochemistry. That is the contribution of Frederick Williams. Berzelius, accordingly, he discovered that lactic acid occurs or is present in muscle tissue, and it is not only present in milk. And please note that your Berzelius here is actually the first individual to make distinction between organic compounds and inorganic compounds, or to make a differentiation between organic compounds and inorganic compounds. Of course, you already know that your organic compounds, they are carbon containing compounds, and your inorganic compounds, they do not contain carbon. Okay, and yes, your Berzelius, he formulated the general principles of catalysts, and he recognized that thiolene in saliva and pepsin of gastric juice are biological catalysts. Okay, so Berzelius is credited with the chemical terms catalysis. Okay, he's credited with the chemical terms catalysis. Of course, when you say catalysis, you're going to speed up the reaction, and then you have polymer, isomer, protein, and some other terms. He is also the one. Berzelius was also the one who coined the term biliverdin. Okay, he's also the one who coined the term biliverdin. Okay, that is for. Then you have Emil Fischer. Emil Fischer was the proponent of your lock and key theory of enzyme action. Okay, he was the proponent of your lock and key theory of enzyme action. This lock and key theory of enzyme action, we will take a look at this one in detail as soon as we move to enzymology. Again, your Emil Fischer, he proposed the lock and key theory of enzyme action. The enzyme being the lock and the substrate being the key. Okay, because in this theory, okay, there should be the key, okay, the key should be complementary for the reaction to occur, or the substrate should be complementary for the enzyme so that the reaction will occur. Okay, take a look at the lock. Okay, you need to have the correct lock. I mean, you need to have the correct key to open a lock. Anyway, Emil Fischer, aside from this lock and key theory of enzyme action, he discovered the name, I mean, hydrazines. He discovered hydrazines. And yes, okay, also there is this Fischer projection in writing chemical reactions. That is for the contribution of Emil Berg here discovered the enzyme pyruvate decarboxylase, or during his time, he just called it as carboxylase. Again, Carl Neuberg discovered the enzyme pyruvate decarboxylase. And of course, this enzyme, this pyruvate decarboxylase, catalyzes the decarboxylation of pyruvate to produce acetate. And yes, Carl Neuberg here also formulated a theory for the alcoholic fermentation of glucose. And yes, Carl Neuberg introduced the term biochemistry in 1903. And aside from those, he was actually, uh, he has a contribution in the making or in the production of gunpowder or in the production of ammunitions somewhat. And with all his experiments, with all his contributions and experience, he was able to contribute to the field of biochemistry and pave the way to a better understanding of metabolic pathways. And that is for your Carl Newton. Then you have here the study or the contribution of Oswald Avery, Colin MacLeod, and Maclean McCarthy, wherein these individuals uh, research on the transforming principle, okay, of Griffith. How did they do that? They what you call this? They extracted DNA from a toxic strain of Streptococcus pneumoniae and mixed the DNA with a non-toxic strain. And accordingly, they observed whether transformation or conversion to the virulent Streptococcus form still occurred. And this study, the study of these individuals, provided actually a strong evidence that genes are made of DNA and not made of protein. Again, the study of these individuals provided a strong evidence that genes are made of DNA and not made of protein, which is okay supported by the study of this James D. Watson and Francis Crick. Okay, again for your Oswald Avery, Colin MacLeod, and Maclean McCarthy, this stated, okay, in terms of DNA, they said that DNA is the substance, okay, the oxyribonucleic acid is the substance that causes bacterial transformation. Okay, and then you have James D. Watson and Francis Crick, who discovered the DNA replication process, as well as the helical structure, the double helix structure of your DNA. And yes, this DNA replication process transmits biological information to the succeeding generation. And then, of course, you have here your central dogma for molecular biology. It states that the information that is encoded in the DNA into the oxyribonucleic acid is transcribed to RNA and then translated into a protein. So you have here transcription and then translation accordingly translated into a protein. And yes, the end product actually for this one is a functional protein. Okay, and please note that the central dogma here is significant in understanding protein synthesis and yes, interrelated to cell function. So I guess let's end here. Let's continue some other time. And this has been Gael, reminding you to live fully, love deeply, love often,
Keep learning and never forget to enjoy the ride. And as you move forward, may you always choose curiosity over fear, kindness over convenience, and growth over comfort. Celebrate your small victories. Honor your quiet moments and trust that every step, no matter how uncertain, shapes the story you are meant to tell. The world is wide, your time is precious, and your journey is uniquely yours. So go make it extraordinary. Again, this has been Gael. Thank you. Thank you so much, and see you around.